Now we are going to talk about transcription in eukaryotes. When we talked about transcription earlier, we had discussed primarily about the prokaryotes, and in that I told you that there is only one RNA polymerase that single-handedly handles whatever is being carried out at the transcriptional level. Also, I told you that there is no compartmentalization and the messenger RNA is directly formed. Okay. In the case of eukaryotes, there is a little bit of complexity. Firstly, messenger RNA is not directly formed. Instead, it is heterogeneous nuclear RNA that is being formed. hnRNA has to be converted into mRNA, and that process is carried out keeping in mind that splicing is to be done and additional stability is given. Okay. One thing is this, apart from this, there is not one RNA polymerase, instead there are 3 RNA polymerases. Okay. RNA polymerase 1, it is responsible for synthesis of 28S, 18S and 5.8S. Now, can you make it out what it would be? Because S is there, sedimentation coefficient is there. This is ribosomal RNA. So, RNA polymerase 1 in eukaryotes 1 is responsible for formation of ribosomal RNA. Like that, RNA polymerase 3 is responsible for transcription of tRNA. Okay. Apart from that, it is responsible for small nuclear RNA transcription as well. And the RNA polymerase 2 is responsible for formation of hnRNA. So, we see functional specialization of different enzymes over here. We are concerned with converting this hnRNA and get mRNA, because that is the resultant product that we aim at when transcription is being done. So, how do we obtain mRNA? First thing is that this heterogeneous nuclear RNA contains introns and exons. Remember, in the previous lesson we talked about what exons and introns are. Exons are the important ones, introns have to be removed. Now, how they will be removed? They will be removed by the process of splicing. Okay. Splicing is done in spliceosomes. How it is done? Now, supposedly you get this mRNA. hnRNA is here. Firstly, at the 5' end, you are going to add a special type of nucleotide that is known as methyl guanosine triphosphate. It would be added at the 5' end, and substrate not being substrate specific, there is polyadenylation. First is addition of this, and the second step is polyadenylation, which gives it additional stability, which gives the additional stability to hnRNA. This polyadenylation is done without taking into consideration what substrates are there. There are many adenyl groups which are added at the 3' end that leads to the formation of poly A tail at 3' end. While here we add methyl guanosine triphosphate, here we have poly A tail. Now, this is being added over here, this is being added over here, and this enables the spliceosomes to come and attach at the intron positions. Now, supposedly this is the intron position, spliceosome comes and joins over here, then here, and they just take the introns away like magnets. That is known as splicing. Now, what you are left with? This part is gone, this part is gone, this part is gone, and this is gone, and what you are left with is all exons, and you have the required messenger RNA in your hand. Okay? So, this is what we have as additional mechanism that is found in the eukaryotes. Now, one thing you have to keep in mind is that this arrangement of introns and exons is somehow pointing towards the evolutionary mechanism. Why introns are present in between exons if they are not required? Maybe with time we as eukaryotes, or simply the eukaryotic cell, learnt that this part is not required, it is better to remove it, because ultimately this thing is pointing somehow towards that this is still an RNA world. Though DNA contains the information, RNA dominance is there, that RNA is extracting only the required information. Out of the all information, huge amount of information that lies in the DNA, only small part of it becomes the transcription unit. Even in that small part, only the exons form the mRNA, which would later on form the proteins. So with this we come to an end of transcription. Our next topic will be this mRNA that is obtained, that will take the help of tRNA and bring to its dispense small amino acids, join them and form the proteins.